Good evening. This is chapter 9, heredity. So, heredity is something that is transmitted in the form of genetic characters from our parents. Nama parents kitenda offspring. So, this is the characteristic features transferred from the parents to the offsprings. Through what? Through a genetic material. What is this genetic material? This genetic material is also called the gene. This gene is in the form of chromosomes, chromatin threads, and the substance that codes for this is called the DNA. So any characteristic is coded by means of deoxyribonucleic acid in the form of triplet codons. We call them triplet codons, and they are the ones they determine the characteristic feature of an organism. So what is heredity then? Is it always the parental characteristics that comes to the progeny? It could also be the grandparents' characteristic features. Say, for example, dog's fur coat color comes from either of its parents. They might have inherited from their parents. So from one generation to another generation, this inheritance of characteristics occur. So these characteristics are also called traits. Okay. So these inherited traits can be controlled by the genetic material. So what is evolution? So if heredity is inheritance of characters from their parents through genetic material, what is evolution? So evolution is change in certain characteristic features over a period of time. Evolution is nothing but this characteristic that is usually transmitted from the parents to the offsprings. There will be some changes. So this occurs over a period of time. Okay, as the time passes, according to the environmental condition, certain changes occur in the characteristics being passed from the parents to the other generation, that is changing. Sila changes occur, that is called evolution. Say, for example, they were monkeys. So the monkeys had tail and they walked in 4 legs. But what happened is, over a period of time, what happened? They started losing their tail and they started walking straight in 2 legs. So this is evolution. So the change in the characteristics of a species over several generations. So changes over several generations. So it will take several generations to change the characteristics. Several generations it will take. For what? For acquiring some differences. Okay, there will be some changes in the same species. This will occur. So this is called evolution. So this is what we are going to learn in this chapter. So what is heredity? Heredity is nothing but we are acquiring characteristics from our parents. So these characteristics can be either acquired or it can be through genes. Okay, so when it is inherited by genes, so parents give rise to offsprings of the same kind. So either the father's character or the mother's character is transferred to the child. So this transmission is done through genetic material or DNA. This is done by reproduction. So sexual reproduction again tends to show off lot of variations because they go about different combinations. So there will be different combination because there is a process called crossing over occurring, and also there will be two parents involved. So both their characters will combine. So there will be change in the characters of the offspring. So this is heredity, but still in this, the traits that are inherited, so the traits or the characteristics can be either inherited or it can also be acquired. So normally these inherited characteristics, it is controlled by genetic material called DNA, which is inside the chromosomes. Okay. This inheritance only is transferred from one generation to another. But this acquired traits, there are some acquired traits. Say, for example, discipline, good characteristic features. These are all acquired from our environment. I could have been born with thick black hair, but when I move to some places where it is where only chlorinated water or salty water is available, I may lose my a texture of the hair. So that will be an acquired trait. It is not coming by birth. Okay. Reproductive processes are divided into asexual and sexual processes. In asexual processes, exact clones are produced, but still they may have some error during copying of genetic material, due to which there will be changes. Also, environmental factors can be responsible for bringing about variation in asexual reproduction. In sexual reproduction, variations again can occur in sexual reproduction also. The reason could be two parents are there, so either of their characteristics will be received by the offspring, some traits from one parent, others from the other parent. So there will be different combinations and permutations. So if the sexual reproduction involves interbreeding, then also the organism that is resultant could be little different. So there are horses combined with donkeys. We produce new species. Okay, new organisms are produced. This here is an exact example for reproduction. This mother giraffe has produced an offspring similar to the parent. Here, this is a haploid sperm cell. This is a haploid sperm cell, and this is a haploid egg cell. They have n number or haploid number of chromosomes, which when combined to form a diploid cell to n number, and they undergo subsequent mitotic cell division, resulting in the offspring, which will have the traits of both the male as well as the female parent. They will have resemblance of their parents. Okay, so this is the cell, and this is the nucleus of the cell where it holds the genetic material. So this is a double helix of a DNA. These are the deoxyribonucleotides that code for a particular gene. So here we are going to talk about what is variation. So how does variation occur, and why does variation occur at all? Why do we need variation? Darwin has an evolution, says that as evolution happens, in the process of evolution, nature tends to select. This is called natural selection. So according to the environment, a species undergoes so many changes. So what is the use of variation? Variation will help the organism to adapt to the new environment, So All of a sudden there could be a change in temperature, there could be global warming. Slowly the species has to evolve. Okay, so for this adaptation to the new environment, variation is very important. This is the starting point of evolution. We need some a start point for evolution. Evolution takes place through many generations, but it all starts with a small variation. If the variation is a suitable type of variation, it is getting adapted to the next generation also. It is transferred to the next generation also. Okay, under adverse condition, it will help the organisms to survive. Okay, so new traits are getting formed. So we find a small finch here, and the beak is getting modified according to the food availability around. Fruits சாப்பிடுறதுக்கு அதோட beak இப்படி modify ஆயிருக்கு. Seed eat பண்றதுக்கு it is getting modified in a different way. For crushing the leaves, their beak are getting modified into different forms and shapes, and slowly this natural selection takes place, and the organisms show a whole lot of variations. Variations are created in the organism according to the environmental condition. So here we will see a few things about the meiotic divisions in order to understand how the variations happen in the subsequent generations. So starting with a pair of homologous chromosomes, what happens is the chromosomes initially duplicate. This is meiosis. Okay, for meiosis division, it is getting prepared. The chromosomes are getting prepared. In meiosis 1, we have prophase 1. In prophase 1, what are all the various phases found? So we find leptotene, zygotene, pachytene, diplotene, and diakinesis in all prophase 1. Okay, here haploid cells with duplicated chromosomes are there. Do we see here? There is one. Here there is one. It is getting duplicated here. We are ending up with 2 haploid cells, but they have duplicated chromosomes. So here that was 2n number. This is the parental cell, but this is having it is appearing to have 2 chromatids, but it is actually a duplicated one. This is n number. Again in meiosis 2, it is not going to duplicate. In meiosis 2 also I have prophase 2, but here it will not duplicate. Simply they will align themselves and start separating, ending up in purely n number of cells. So this is what we are having. Those ova and sperm cells, they tend to contain only n number of cells in their gametes. So gamete cells are always holding n number of cells. So clearly we can look over here. So the sister chromatids are the blue ones. The blue one and the red one are non-sister chromatids. Initially, you find that the sister chromatids are duplicating. Okay, you have the sister chromatids which are going to duplicate. So in the leptotene, the chromosomes condense, and in zygote synapses begin. They slightly touch over each other. Okay, then the crossing over will occur in the pachytene stage. You can see the end product of crossing over, wherein these are totally red chromosomes. Here you have partly blue ones transferred to the red chromosomes. So in pachytene stage, the DNA exchange is occurring between the non-sister chromatids. The blue ones and the red ones are interchanging the chromosomes. This is why there is variation in the progeny, in the offsprings. That is why we have a whole lot of variations. So in diplotene, you have the chiasma visible, and they also become, okay, in the bivalence, you have, these are all bivalents. Okay, they have duplicated, no? So in the tetrads, these tetrads, they go, these are tetrads. Okay, 2 of them have got attached, and they are getting lined up in the midline. Okay, now what happens is they are getting separated. So after crossing over, after recombination of genes, what happens is in the metaphase 1, the tetrads are attached to the spindle fibers at their centromeres. Centromeres are here. So these are the spindle fibers that are getting attached to the centromeres and they are getting separated. In early anaphase, they are getting separated. And the paired chromatids move along the spindle fibers and they reach the centriole portion. And then what happens is the nuclear membrane will disintegrate and the new cell membrane has to form in between. Okay. So we are ending up in two cells. So at the end of meiosis 1, you have two cells, but all of them have duplicated chromosomes. Duplicated ஆயிருக்கு, recombine ஆயிருக்கு. Characteristics have got interchanged among the non-sister chromatids. After this, what happens in metaphase 2 is what happens? The chromatids again line up at the mid of the cell and the centrioles pull them along this side and that side. Okay. Here again duplication will not occur. As such in the ஏற்கனவே இருக்கிற cells ரெண்டா பிரிச்சிருவாங்க. ஓகேவா? So that is why we are ending up with n number of cells. So we are ending up with 4 haploid daughter cells getting formed at the end of telophase 2. Okay, we have 4 cells created from the parental cells. So now we understand from where this variation occurs. So this is a mom, this is a father. So this is their female gametes and male gamete. So they are getting produced. So you have an egg and a sperm, which the gamete cells, they fuse together and you have a diploid zygote here. So ingeyoda meiotic 2 mudinjirum. Okay, fertilization is fusing of the sperm cell and this egg cell in the n number of chromosomes will fuse with this n number of chromosomes to produce 2n number of cells and subsequently they will undergo mitotic division. Okay, so subsequently we end up with mitotic divisions. So here the parent cell is 2a. So the difference between meiosis 1, 2 and mitosis is shown here. So in mitosis, what happens is they start with diploid number of cells. All the diploid number of cells, get duplicated. They line up in the midline and they separate and give rise to 2n chromosomes in each cell. Okay, the chromosomal number doesn't change. Here in meiosis, what happens is this is 2n number. They duplicate, they form tetrads, they separate, and then what happens is they have duplicated n number of cells. Then each one of them separates to 4 individual n number cells. So this is asexual reproduction. This can be compared to binary fission also. So a cleavage furrow is created right at the center and they separate into 2 daughter cells. So this is what happens in the case of binary fission or asexual reproduction. So this is again a detailed representation of how the plasma membrane separates, resulting in 2 daughter cells. So This occurs in the case of plant cells. In plant cells, the difference is it has to get a cell wall formed in between. So after duplication of vesicles, chloroplasts, and all the cell organelles in a plant cell, it has to form a cell wall in between the cell also. Okay. Now, getting back to the chapter, what did Mendel do in his experiments? So what did Mendel do in his experiments? We will see now. So he started with a pea plant. What is a pea plant? Patani chedi. Okay, wow. So pea plant is a self-pollinating plant. Self-pollinating na enna appadina அதுவே அதோட pollen grains வச்சே வந்து it will be capable of bringing out a fruit, peas. Okay, so what happens is the pollen grains fall on its own ovules to generate peas. Okay, but what he did was he was extracting the anthers manually. Manually, வேற ஒரு பிளான்ட்ல இருக்கிற ஆந்தர்ஸை எக்ஸ்ட்ராக்ட் பண்ணாங்க. எக்ஸ்ட்ராக்ட் பண்ணி என்ன பண்ணாங்க? எக்ஸ்ட்ராக்டட் ஆர் எக்ஸைஸ்ட்னா கட் பண்றது. ஓகேவா? எக்ஸைஸ் பண்ணி என்ன பண்ணாங்க? இன்னொரு பிளான்ட்டோட சேர்த்தாங்க. So if you are taking the female part from this cell, see, I'm sorry. So this is removing the stamens from this one. Excise means remove. All the stamens are cut. So what happens is from the white flowers, he is going to transfer the pollen grains to the ovules of the violet one. So violet ka white da kudukudhu. Okay va? Transfer the sperm bearing pollen from the stamens of white flower to the egg bearing carpel of purple flower. So purple flower oda ovules ka in the இடத்துல it is transferred. Then what happens is they mature into pods. Okay, pods are created. So what happens in emasculation? We call this process as emasculation. E-M-A-S-C-U-L-A. This is emasculation process. Okay, so what happens is they are manually. So this is a bisexual flower. We should remember that it has both male and female parts in the same flower. It doesn't need another plant or another flower to pollinate itself. It's a self-pollinating bisexual flower. Okay, so that is why it is, you have to excise the male part. So the male part வந்து இந்த பிளான்ட்ல இருந்து எக்ஸைஸ் பண்ணியாச்சு, கட் பண்ணியாச்சு, ரிமூவ் பண்ணியாச்சு. So that is going to be that is going to be interpollinated by the other one. So what happens is this is the parental generation. In the இதுதான் வந்து parental generation. So this is the F1 generation, first filial generation. For first filial generation la patani produce ஆயிருக்கு. We are waiting for the carpel to mature into pod. What you are going to get is all or violet flowers, because the gene that is coding for violet flower is a dominant gene. அது dominant gene இருக்கிறதுனால எல்லா progeniயும், அதாவது first filial generationல எல்லாமே வந்து violet colorல தான் இருந்தது. So if it is a dominant gene, then all the this is called a pure breeding plant. Pure breeding plant அப்படினா என்னன்னா ரெண்டு ஜீனுமே. So we call them alleles. Okay, so allele னு சொல்வாங்க. There will be 2 alleles for every gene. Okay, say for example the allele representation I am using it as P for purple color. Okay, so this will be both capital P, this will be both small p. So the first filial generation will be capital P, small p. So what will be the second filial generation? We will have 3 phenotypes expressing purple flowers, out of which only one will be dominant PP. The others will be what? They will be heterozygous in condition. Idoda genotype pathina, either heterozygous arkum. One dominant genavum, one recessive genavum. So this will be both recessive. Recessive genes homozygous condition da express agum. So homozygous condition is a condition where both the genes are recessive or dominant. Homozygous condition la rendu allelume dominant avo illa recessive avo da irukku. So what is homozygous condition? They have similar alleles. Okay, either both should be dominant or both should be recessive. So similar alleles for a particular trait. Say for example, this is purple color, means both the allele will code for only purple color. Okay, ma? For a particular or a specific trait. Okay, so it can either be dominant or be recessive. Okay, so heterozygous is a condition. In a heterozygous condition, what happens is it will be dissimilar alleles. So alleles will be dissimilar. There will be dissimilar alleles, and one could be dominant, the other could be recessive. In this case, capital P and small p denote the heterozygous condition. So capital P is one allele and this is another allele. Okay, what is an allele first? So there will be a pair of genes. Okay, not a single gene determining a character, because one is coming from the male parent, another is coming from the female parent. So allele are a pair of genes, and this will be present at a particular loci. Okay, locus means a particular location. Okay, at a particular location only. In the, say for example, this is your chromosome. This is your chromosomes, that which has not got duplicated. Okay, say for example, so if this is the particular loci for my hair color, then I will have two genes, two alleles, one from my mother and one from my father. So whichever is dominant will get expressed. So alleles are pair of genes. Always there will be two genes, a pair of genes that occupy a specific location. Okay, location in a particular chromosomes. This is my chromosome. Okay, so this is my chromosome and control the same trait. So H also controls hair color. This H also controls the hair color, but they code for different expression of the same character. And the character expression வந்து வேற மாதிரி இருக்கும். Character வந்து hair color தான். Hair color is the character. The expression may differ. That is what they say. Okay, allele definition is this.